Welcome to this short lecture on arbovirus emergence and the vector-to-host molecular switch. My name is Pierre-Yves Lozach, and I'm giving you this roughly 30 minutes lecture. So what arboviruses are? Arbovirus stands for arthropod-borne virus. It's not a taxonomic name, simply because it's a supergroup of various viral families. This supergroup encompasses more than 30 viral families in totals: Reoviridae, Rhabdoviridae, Togaviridae, Flaviviridae, and bunyaviruses. That is an order that by itself includes more than 20 viral families. These viral families include more than 500 isolates together. Many are pathogenic in humans and livestock or wild animals. They share the genome nature, that is a RNA molecule. It can be double strain or single strain. This genome replicates exclusively in the cytosol. However, otherwise, they are very diverse in terms of morphology, structural organization, shape, length, size, and some are enveloped, like most of them actually, and Reoviridae are not enveloped, for instance. Many of these viruses are distributed worldwide. You have a couple of examples on the right. You probably all know dengue virus, West Nile virus, but also Zika virus that has emerged in the 2010s. And many are coming from the bunyaviruses orders, like Crimean Congo hemorrhagic fever, Heartland virus, Rift Valley fever virus, and many others. At the size of the arthropod-borne virus, the virus are actually transmitted from one host to another by arthropod vectors. Usually arthropod-borne viruses, they are maintained in arthropods. There are many arthropods that can transmit arboviruses. Insects like mosquitoes, sunflies, midges, etc., but also arachnids like ticks. And they infect a very wide diversity of vertebrate hosts, including mammals and humans. Usually humans are dead-end hosts. If these viruses are transmitted during the blood meal of infected arthropods, during pandemics, epidemics, they can also be transmitted by aerosols. And the people who are at the higher risk are the healthcare personnel or butcher, people working with people that are infected or animals that are infected. And they can be in contact with the blood of these infected patients or infected animals. And the natural transmission is mainly through the transmission by arthropod during their blood meal. So incoming viruses are are introduced in the skin dermis of host, vertebrate host, or mammalian host, human host, and they can either infect local cells, they can jump into the blood flow, or they can also infect some human cells in the skin dermis. Typically, dermal dendritic cells that are present in the skin dermis are proposed to be sensitive to the infection by many arboviruses, and the arboviruses could use those cells to spread throughout the host, because those cells they can recognize foreign antigens, and once they are activated upon infection, for instance, they are mobile and they will move to the secondary lymphoid organs, where they will activate the immune systems. And the arboviruses could take advantage of this to spread throughout the host. So one fundamental difference between arthropod vector and the mammalian host is that in arthropod vector, infection doesn't show any obvious clinical sign. That means arthropods are infected, but they don't, in appearance, develop symptoms or pathologies. And this is something that we can also observe in culture cells and in cell monolayer. If you infect tick cells with Unraveling, it's an example I decided to show today. Unraveling is a virus, a tick-borne virus. It has never been associated with any disease in humans. However, it's used as a surrogate for highly pathogenic emerging viruses transmitted by tick-bornes, such as Heartland virus in North America or Arbovirus in China, and since it has been discovered, emerged in 2009, is now spread all through Asia, and it's present in South Korea, Japan, and so on. And so when you infect tick cells with Unraveling, after 10 days, 30 days, 2 months, 6 months, you can still collect supernatant and get some infectious progeny. The cells are persistently infected. It's a persistent infection, and the cells continue to divide. They are perfectly fine. On the other way around, in contrast, when you infect mammalian cells, as you will see in the next video, the HeLa cells serve as mock control without any virus. So the cells, you can appreciate that they divide, they are perfectly fine, they are very nice. However, you infect HeLa cells with Unraveling, the cells, after a certain time, they start to blow up, to shrink, and that is typical from cells that are dying, that are apoptotic. When we look at the particle directly and try to assess the infectivity of the virus when it is produced from arthropod vector or from mammalian host cells, we observe typically using these techniques based on dynamic light scattering. It's a free label approach to visualize particle. It's a bit similar to what you are in use when you analyze the FSC and SSC parameters from the flow cytometric assays. You can visualize the particles. And the particles, they can be quantified with these techniques. You can also access to the size of the particles. And when we do so, we found for Unraveling typically a total number of particles per milliliter of 10 to the 11. And that includes the non-infectious particle and the infectious particles. When we compare this number to the infectious particle that is usually determined by Foci forming unit assay for Unraveling, because it's not lytic, but it's also determined by plaque assay, typically for viruses that are more aggressive and lytic. We observe a ratio between infectious and non-infectious particle of 1 to 5,000 particles. It sounds maybe low. However, it's very usual for arboviruses, such a ratio of infectivity. Now, if we analyze the particles produced from tick cells on the right panel, in that analysis, we use SDS page and comma-C staining to look at the level of N protein. The N protein, it's the nucleoprotein, it's present in the viral particles, and we can assume that the quantity of N proteins correlate with the total number of particles. And you can see on this comma-C staining that to reach a number of 4 per 10 to the 7 infectious particles, you need 10 times less and proteins. So that means 10 times less particle, suggesting that the ratio of infectivity when the virus is produced from tick cells is 10 times higher than that from the virus produced in mammalian cells. So the question is how a virus, the same virus, can display a so high difference in infectivity when it is produced from one cell species, cell type, against another. And we need to look at what happens at the molecular level and the cellular level. As we introduce arboviruses, they can replicate in both arthropod vector and mammalian host, vertebrate host. And in a sense, they have a very complex dual life cycle in both vector and host. However, the biology of host and vector differs at large in many aspects. So the carbohydrates, the lipids, but also the molecular machinery to replicate nucleic acids, the cellular protein that could be packed in the particles when they are produced from vector or mammalian host. They can really have a significant influence on the very first step of infection of target cells. It's typically what we will see in the next slides. I pick up a couple of examples to illustrate all of these aspects. And a novel example is Rivale fever virus. It's an endemic virus in Africa. It's highly pathogenic in both human and livestock, and it now represents a risk of introduction in Southern Europe and in Asia. So this virus has a three-segmented genome, and one of these segments, the M segment, when it is replicated and expressed in mammalian cells, encode three proteins. One non-structural proteins, NSM, on the top, and GN and GC, two structural glycoproteins that are present at the surface of the viral particles. When the virus replicates in mosquito cells, because it's a mosquito-borne virus, it can produce an additional polyprotein that is further processed and cleaved in the ER and Golgi apparatus. And it produces not only GNGC and NSM, but another protein, another product that is GP78. In a sense, it's a merged protein between NSM and GN. And that means when the virus is amplified in arthropod vector, it comes with an additional glycoprotein at the viral particle surface. And this can influence at large the virus binding, because most of the interaction between viruses and target cells occur through cell surface receptor and the glycoprotein or the protein present at the surface of the viral particles. And you can easily imagine that it confers the possibility to the virus to have more interactions with different receptors when GP78 is expressed. But also it can influence the signalization, because very often when you interact with the cell surface receptor, you will trigger different signalization depending on the nature of the receptor, the number of the receptor, or the identity of the receptor. It's a first example. And this, it's also has been shown for, documented for now a couple of viruses. When the particles are produced from arthropod cells, they display a significantly different morphology than the viruses produced from mammalian host cells. Here I choose another virus as an example, germistan virus. It's a virus from the Bunyaviruses family. You probably know in North America, lacrosse virus or California encephalitis virus, they belong to the same family. But you can also find in South America Oropouche virus, in Europe Schmallenberg virus, and so on. And germiston virus is used also as a surrogate for all these highly pathogenic viruses in humans and also in wild animal and livestock. And when we look at the particle produced from mosquito cells, for instance, germiston virus, the particle, they look very heterogeneous in size, in shape, in morphology. And in BHK21 cells, mammalian cells, the virus particles that are produced from those mammalian cells, they look much more homogeneous in size and in morphology. It's something we can also determine measuring the circularity of the particles. We measure the width of the particles, and when the index is close to 1, it's a perfect cycle, but lower is the value, and it's more heterogeneous shape that is obviously appearing in the particle stock. And it's something also that we observe for Oconivirus on the very right panel. The circularity is less important when the particles are produced from tick cells than the virus stock produced from mammalian cells. The size also is significantly lower. Of course, here you could say it's only 5-6 nanometers difference, but these 5-6 nanometers in terms of structural organization of the particle, they are dramatic. And the particle size, the particle shape also influence the pathway that the virus can use to infect the target cells. Because usually the endocytic pathway, because in that case, these viruses are endocytosed when they infect target cells, they also depend mainly on the size of the cargo they can uptake. Now, we talk a lot about virus binding at the cell surface, but a virus, once it has attached to the cell surface, it has to cross a certain number of barriers. And these barriers, they can be extracellular matrix. Very often it's carbohydrates, such as heparan sulfates. They can be lipids and proteins, membrane proteins, transmembrane proteins, also protein associated with the plasma membrane. And the lipid, it can be simply also the plasma membrane that represents an important barrier for viruses to access the cytosol. And finally, they can be also the cytoskeleton, which is usually mainly composed of actin, for instance. And the particle attach in many cases through unspecific interactions, unspecific attachment. And once the number of particles is dense enough, large enough, concentrated enough at the plasma membrane, they can interact with the specific receptors. And two options are offered to the virus. Either it fused at the plasma membrane if it's another virus, or it is sorted into endosome. It's another possibility. For non-enveloped virus, it's different mechanisms. It's not fusion, of course, because there is no envelope. They will perforate the membrane with some viral proteins at the surface of the particles. Sometimes it requires additional factors, co-receptors, cellular factors that ensure the internalization, or they are also important for the activation of fusion of the particles. And all together, it provides even more possibility for the virus to differ in terms of early virus or cell interaction, depending on the cells from which it originates. So that means, again, arthropod cells or mammalian cells. All of these interactions, they determine the species that the virus can infect and also the tissue tropism. And I choose another example, a recent example of a receptor that has been recently identified for Rivallet fever virus, LRP1. LRP1 is a protein of the LDLR receptor families, and it has been shown it can interact directly through protein-protein interaction with the GC protein. But again, in that context, if you think about the virus, it's something we discussed previously. Rift Valley fever virus, when it is produced from mosquito cells, has an additional GP78 that could interact with a different receptor than the particles produced from mammalian cells, for instance. And again, it offers a broader diversity of interaction, possible interaction for the virus. So since the LRP1 has been identified for as a receptor for Rift Valley fever virus, many other viruses have been listed with the capacity to interact with that receptor through protein interactions. Not only protein-protein interaction occur in the various receptor interactions. It can be also interactions between glycans and cell surface receptor. And again, the glycans produced in arthropod vector are very different from those produced in mammalian host. Typically, in mosquito cells, in insect cells, usually the glycan, the carbohydrates, are heavily manosylated, whereas in mammalian host, the glycan are still using as a basis the mannose residues, but then there are many modifications in the ER, in the Golgi, leading to much more complex glycans with much larger diversity of glycans in the carbohydrate structures. And here it's again the example of Ukunimi virus. When the virus is produced from tick cells, the electromobility of GN is much faster when the particles are produced from tick cells than when GN is analyzed from particles produced from mammalian cells. When we use some endoglycosidases like PN-gas F, PN-gas F specifically cut all glycans linked to the N-asparagine in glycoproteins, we observe that most of glycan, if not all, on GN produced from particles produced in mammalian host, they can be removed with PN-gas F, as you can appreciate on the right panel. When the particles are produced from tick cells, the GN is nearly not sensitive to PN-gas F treatment. So either there is no glycan on the glycoprotein GN, or the glycan are not sensitive to PN-gas F. So you think that they are really different. And if you can also not notice here on this right panel that the electrophoretic mobility of GN is much faster when the particles are produced from tick cells than mammalian cells without any treatment. And here we can say that there is no cleavage, no deletion in the gene coding for GN or the proteins. It suggests a different folding of the protein and probably also different glycans. And why I say it's important, it's because many virus receptors in mammalian hosts in humans have been linked to the lectin family. So lectins, it's a huge family of protein that can recognize glycans, carbohydrates of different types, different nature, and many of them can serve as receptor for arboviruses. One of them, which is important, it's DC sign, because DC sign is a C-type lectin that is able to recognize specifically mannose residues. So mannose residues are typical of the glycoproteins produced from insect cells, notably mosquito cells. And why it's important, it's easy to understand, to envision. It's because the incoming particle, when they are transmitted by the infected arthropod during the blood meal, they come therefore from arthropods, mosquitoes, and they come with a dense glycosylation in mannose residues. And these mannose residues, they can be recognized by DC sign. This is mainly expressed, highly expressed in dermal dendritic cells that are located in the anatomical site of transmission. It's the reason DC sign is believed to be particularly important for the very first step of infection by arboviruses. But again, if we put that in the context of this comparison between virus produced from arthropod vector versus mammalian cells, the particles produced from mammalian cells come with a different glycosylation and potentially cannot interact with DC sign, while the particles produced from mosquito cells exclusively interact with DC sign. It's another important example. Not only glycoproteins of the virus can contribute to target and attach the cells of host. Here I choose this example of what we call viral apoptotic mimicry. Many arboviruses, notably dengue virus, West Nile virus, chikungunya virus, or even some they are not arboviruses, but still these viruses, they embed phosphatidylserine, which is a marker of apoptotic cells. I won't enter into the detail of the mechanism because it's still debate and it's also not completely well understood. But the model, the hypothesis is that the particle can embed phosphatidylserine during the virus assembly and production, and the particles are released with this phosphatidylserine that can be recognized on target cell by cell surface receptors like the TIM receptor family or AXL and TIR03. And it promotes the virus attachment to target cells. And again, for instance, there is an important difference in the production of phosphatidylserine between insect cells and mammalian cells. There are other glycan and there are other factors on viral particle that can contribute to the attachment of the variants, the particles to target cells. And for instance, my group has recently identified a glycolipid, the glucosylceramide, that is enriched in virus particles, in the envelope of the virus particles. And these glycolipid contribute to the attachment of the particle to target cells. And all of these factors, the lipids, they are also important not only for the attachment, they can be also important for the subsequent step of the fusion for envelope viruses. The fusion, it's the ultimate step of the virus entry program. It can have a role simply because it make it more or less easy depending on the lipid composition in the envelope particle. The composition in the target, the composition of the lipids in the target membrane is also important because, as I said before, the lipid composition is different in arthropods from mammalian host. And cholesterol or even also glucosylceramide has been involved, and there are some differences. For instance, insect cells are not able to metabolize the cholesterol. They need to take the cholesterol from the medium, the feeding medium. And while mammalian cells, they produce, they can synthesize this lipid. And this lipid is also believed to be important for the fusion mechanisms of arboviruses, specifically then, therefore, in mammalian hosts, but less probably in insect cells. It's completely different for non-envelope viruses like Reoviridae, because of course they have no envelope and so they don't fuse, but they use different mechanisms. I won't enter into detail here, but we can also easily imagine that depending on the cells used to produce these viruses, we can also impact the proteins that are present at the surface of Reoviridae, and it can influence the mechanism by which they will create a pore in the target membrane to deliver their genome into the cytosol. At the molecular level, we have illustrated and discussed many of the factors, the compounds that can influence the early host cell interactions and viruses. And there are additional facts that can emerge from these molecular differences between viruses produced from arthropods and mammals. We have briefly mentioned possibly the signalization that is influenced through the interaction between the virus and its receptor. And this signalization is important for the internalization of the envelope virus into the cells, for instance, but it can also have impact on the immune system of the host cells to activate the immune system. On the other way around, we could imagine that viruses, as early as the virus entry steps, can modulate the host cell machinery, the cell stress responses, for instance, to prepare to create a propitious environment for its own replication. And that it can happen at different steps during the entry process. Typically, the most relevant for these differences in terms of molecular composition in the different virus stocks produced from mammalian cells and arthropod vectors. You can typically believe and imagine the toll-like receptor in endosomes, for instance, that recognize also incoming pathogens, depending on the nature of the composition of the particle. The activation and the subsequent signalization can be different. But also, after the entry, after the delivery of the genome of the virus into the cytosol, the genome, for instance, can be edited in mammalian host. It can be different in insects. We can think that genome editing, the methyl group that are present or RNA of the virus, they are again different if they are coming from mammalian cells or arthropod cells. The post-translational modification, ubiquitination, phosphorylation, and so on are also some possibility of differences between the virus produced it's in vector cells or in the host cells. And all together, they can influence at large the spread of the virus, because very often it's matter of few minutes. It's a race between the host cells, where the immune system will be activated and react, and the kinetic of entry and replication of the virus. If the virus is fast enough or too fast, the immune system doesn't take over and the virus wins. However, if the immune system is fast enough to react and to counteract the virus infection, it's the immune system that wins. And all these differences, they can sound not so much significant or detailed, but combined together, they can give these few seconds, few minutes to the virus to take over the immune cell responses and to counteract the cell stress response mechanisms. And now to finish with this lecture, I would like to just open what we discussed today on the molecular transition between arthropod vector to mammalian host. We are focused here on the fact that arboviruses come with a coat, which is the arthropod coat when it is introduced in mammalian host. Once the arbovirus has infected the first mammalian host cells, what happens? The arbovirus completely changes the coat. It's different compound. It's the compound from the mammalian host with the glycan, the lipids again, and so on. And it's a completely different envelope coat. But if we think about, let's talk about again skin cells, for instance. Skin cells, they have also some distinction in their cell biology compared, for instance, to liver cells that are very often targeted by arboviruses. So the same concept could be also applied inside a host, a mammalian host. The virus could come with the coat of the skin cells and infect liver cells later during the propagation. And once in liver cells, the virus will again change its completely course. So it has an advantage also for the virus because it's in a way a stealth way to escape, to evade the immune system, because every time it will use the compound of the host and it will continue to change. So we could also see that as an important advantage for these viruses. And again, of course, to close the loop, when the virus is produced in mammalian host, an arthropod will come during the blood meal, will catch this virus with the mammalian coat, and this process, this principle is also true when the virus will infect again, return into the arthropods. So I have now finished, and thank you for your attention. So I would be more than happy to take any questions, and feel free to email me to ask your questions. I will do my best to address all of them. Thank you again for your attention.